Hi everyone, welcome to Latest Trends 2023, the channel where we explore the most exciting and controversial developments in science and technology. I'm Jay, your host for today. In this video, we're going to talk about gene editing, a powerful technique that allows scientists to modify the DNA of living organisms. Gene editing has been making headlines in recent years for its potential to cure diseases, enhance traits, and even create new life forms. But it also raises ethical, social, and environmental questions that need to be addressed. Gene editing is not a new concept. Humans have been altering the genes of plants and animals for thousands of years through selective breeding and hybridization. But what makes gene editing different today is the precision, speed, and ease with which it can be done. One of the most widely used tools for gene editing is CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR is a natural system that bacteria use to defend themselves against viruses by cutting and pasting DNA sequences. Scientists have adapted CRISPR to edit any gene in any organism by using a guide RNA molecule that matches the target DNA sequence and an enzyme called Cas9 that acts as a molecular scissors. CRISPR has been used for many purposes, such as creating disease-resistant crops, engineering animal models for research, correcting genetic defects in human cells, and even creating gene drives that can spread a desired trait through an entire population. But CRISPR is not perfect. It can sometimes cause unintended changes in the genome that may have harmful consequences. It can also raise ethical dilemmas about who should have access to gene editing technology, how it should be regulated, and what are the long-term effects of altering the natural code of life. For example, in 2018, a Chinese scientist named He Jiankui shocked the world by announcing that he had used CRISPR to edit the embryos of twin girls to make them resistant to HIV infection. He claimed that he did it for humanitarian reasons, but he was widely condemned by the scientific community and the public for violating medical regulations and ethical principles. He was later sentenced to 3 years in prison by a Chinese court. Since then, scientists have continued to improve CRISPR and other gene editing techniques, such as base editors and prime editors, that can make more precise and accurate changes in the DNA without cutting it squared. They have also developed more sophisticated ways to monitor and control gene editing outcomes, such as using molecular switches and anti-CRISPR proteins cubed. Gene editing has also been applied to various fields of medicine, agriculture, biotechnology, and conservation. Some of the recent examples include using CRISPR to treat sickle cell disease in patients by restoring their ability to produce normal red blood cells, using CRISPR to create hornless cows that are more comfortable and safer for dairy farming, using CRISPR to engineer mosquitoes that are resistant to malaria or unable to reproduce, using CRISPR to restore vision in blind mice by repairing a genetic mutation that causes retinal degeneration. These are just some of the amazing possibilities that gene editing offers. But they also come with challenges and risks that need to be carefully weighed against the benefits. Some of the questions that we need to ask ourselves are: how do we ensure that gene editing is safe and effective for humans and other organisms? How do we balance the rights and interests of individuals, groups, and society when it comes to gene editing? How do we prevent the misuse or abuse of gene editing technology for malicious or unethical purposes? How do we respect the diversity and dignity of life forms that are affected by gene editing? How do we protect the environment and biodiversity from the potential impacts of gene editing? These are not easy questions to answer. They require a lot of scientific research, public engagement, ethical deliberation, and policy regulation. That's why experts from different disciplines and backgrounds have been meeting regularly at international summits on human genome editing to discuss these issues and provide guidance for responsible governance. Gene editing is one of the most powerful and promising technologies of our time. It has the potential to improve our health, food security, animal welfare, and environmental sustainability. But it also poses significant challenges and uncertainties for our future. We need to be aware of both the pros and cons of gene editing and make informed decisions about how we want to use it. Thank you for watching this video. If you enjoyed it, please like, share, subscribe, and hit the bell icon for more updates on latest trends in science and technology. I'm Jay, signing off for now. See you next time.